Hello everyone, so now this is part two of the chromatography experiment. I don't know if you can see here, I've dried, I end up laying them or hanging them instead of laying them down. I dried the chromatography paper. It absorbed all the way up to the line and I let it dry. So now I'm going to actually prep them to be inside, essentially encased in these glass slides. And if you notice, I've rearranged a bunch of stuff. I got rid of everything I didn't need, and I added all the stuff I need. Acetone, I have the hormone control, I have the actual hormones. And the idea is I'm going to be sandwiching them in between, the paper in between two slides. Now, this isn't any protocol, or at least that I know of, anywhere. I pretty much just came up with this, and I don't know if it's going to work or not. What I'm going to do is I'm going to sandwich them in between and then use some parafilm to seal it. But I'm only sealing the sides of of it, not the top or the bottom. Now, parafilm is a pretty cool stuff. It's actually made of paraffin wax, and it's almost like what Saran wrap is supposed to do. This stuff actually does. So there it is, with a little section hanging out at the bottom. If this does work anyways, I'm going to see if I can refine it into a more, I guess, elegant form, I guess, maybe. So there's the other one here. So now I'm on to the second step, or second protocol. Now I'm going to transfer some hormones from this. Now this is light protected, because it indole acetic acid degrades over time with light, so I have to keep it in here. And I have two microcentrifuge tubes. One's labeled A and one's labeled B. That's for, which will correspond to each paper. Right now I'm just testing to make sure it actually fits in there before I actually mess around in there. It's where sometimes you have, when you're coming up with something, you have to modify it. Right now I'm just kind of testing to see if, before I even put any chemicals in there. I just want to make sure. I'm going to use an extra slide to give it a little separation, and then perhaps I can hang it at once. Yeah, that should be enough. And any sort of modifications like this to the original protocol, you do want to document. That way you know what you've done and what you didn't do. I'll set them right there, and then I'll continue on with what I was doing. I'm going to take the hormone out, indole acetic acid, and then right now I need 1.5 milliliters in the one labeled A. So I'll do that. This stuff smells nasty. I'm trying to think of what the smell is like. Now I'm going to move. I don't know why I did this, but I could have just skipped this step, but that's what I wrote, so that's what I'm doing. I'm going to transfer 500 milliliters to B. Yeah, that was a wasted step. Close A up. And now I'll close B up for a second. And now I'm going to take A or B, and I'm going to add the hormone control, which is it doesn't have any control or any hormone in it, but it has the same solution chemical, which I made a video a couple days ago about how I made it. I'm going to put 500 milliliters in there. Whoops. And what I'm doing is I'm diluting the indole acetic acid by 50%. And now what I'll do is I'll use the vortex mixer to mix it up to make sure it's all mixed together. And I don't know why I'm not using this, but I should just use one of these, make it easier. That's what they're there for. And I don't need that anymore. Now I'm going to transfer 2 microliters on, I'm going to make a 2 microliter dot on each paper, corresponding with A and B. Okay. Now, before I do that, what I'm going to do is I'm going to put some acetone in this beaker here. I'm going to do about 25 milliliters. And then I'm going to put 1 dot of 2 microliter dot on each one, right at the edge here where the glass is exposed, or it's exposed from the glass, right in the center. And this is a really small amount. And it's always important to change your pipette tips when you use a different substance or different concentration of substance. So now I'll get this set up like this, sandwiched together, and I'll put it on the clip. And then now I will lower it into the acetone. Now I'm going to put it in just about a milliliter or a millimeter in, just barely touching. Okay, so what I'll do now is I'll show you exactly what I'm trying to get to happen. If you see here, there's on this diagram, I don't know if you, okay, so this is a slide. Here's the A and B label, and there's 2 lines, and what I'm going to do is I'm going to let it go up to this line and soak up to that line again, or line. And this is the initial line where I let it soak down, where the other compound soak down. So the idea is, if you see this little gray dot, it's going to get sucked up through and hopefully turn red as it goes through the other compound. And essentially that's what I'm going to hope happens. And what I'll do is I'll let that do its thing, and then hopefully I'll let you guys into part 3 where we can see the result.